Hello, my name is Joshua Mugabi. I welcome you to Blood. What are the main functions of blood? The blood is a means of substance transportation throughout the body. The blood distributes nutrients, oxygen, hormones, antibodies, and cells specialized in defense to the tissues and collects residues like nitrogen wastes, carbon dioxide from them. What are the constituent elements of the blood? The blood is made up of a liquid and a cellular part. The fluid part is called plasma, and in it there are several substances like the proteins, the lipids, carbohydrates, and mineral salts. The cellular constituents of the blood are also known as blood capsules. They comprise of the erythrocytes, the red blood cells, leukocytes, and platelets. What is hematopoiesis? Hematopoiesis is the formation of blood cells and other constituent elements of the blood. Where does hematopoiesis occur? Hematopoiesis occurs in the bone marrow, mainly within the flat bones, where erythrocytes, leukocytes, and platelets are made, and in the lymphoid tissue, responsible for the manufacture of leukocytes and found in thymus, spleen, and lymph nodes. In which bone can bone marrows chiefly be found? Is bone marrow made of osseous tissue? Bone marrow can mainly be found in the internal cavity of flat bones like vertebrae, ribs, scapula, sternum, and the hips. Bone marrow is not made up of osseous tissue, although it is a connective tissue as bone tissue is. What are blood stem cells? Stem cells are undifferentiated cells able to differentiate into other type of specialized cells. The stem cells of the bone marrow originate the differentiated blood cells. According to stimulus from specific growth factors, The stem cells are turned into red blood cells, the leukocytes and megakaryocytes, cells that form the platelets. Research shows that stem cells of the bone marrow can also differentiate into muscles, nerves, and hepatic cells. What are the other names by which erythrocytes are known? What is the function of these cells? Erythrocytes are known as red blood cells, RBCs, or red capsules. Red blood cells are responsible for oxygen transport from the lungs to the tissues. Is the name of the molecules that transport oxygen in the red blood cells. The respiratory pigment of the red blood cell is hemoglobin. What is the molecular composition of hemoglobin? Does the functionality of hemoglobin as a protein depends on its tertiary or upon its quaternary structure? Hemoglobin is a molecule made up of 4 polypeptide chains, each bound to iron-containing molecule called hemoglobin. So the molecule contains 4 polypeptide chains and 4 hemoglobins. As a protein composed of association of polypeptide chains, the functionality of hemoglobin depends upon the integrity of its quaternary structure. On average, what is the life duration of red blood cells? Where are they destroyed? What is the destination of the hemoglobins after the destruction of the hemoglobin molecule? On average, red blood cells live around 120 days. Spleen is the main organ where all red blood cells are destroyed. During the red blood cell destruction, the hemoglobins turn into bilirubin, and this substance is then captured by the liver and later excreted in the bowels as part of bile. What are the functions of the spleen? Why is a splenectomy, surgical removal of spleen, compatible with life? The spleen has many functions. It participates in the destruction of old red blood cells. It is specialized leukocytes are matured. It helps to renewal of the hematopoietic tissues of the bone marrow when necessary. It can act as a sponge-like organ to retain or liberate blood from or for the circulation. Total splenectomy is not incompatible with life, as none of the functions of the spleen are vital and at the same time exclusive of this organ. What is anemia? What are the four main types of anemia? Anemia is low concentration of hemoglobin in the blood. The four types of anemia are the nutrient deficiency anemia, anemia caused by blood loss, hemolytic anemia, and aplastic anemia. Nutrient deficiency anemia is caused by dietary deficiency in fundamental nutrients for the production and functioning of the red blood cell, like iron deficiency anemia, vitamin B12, and folic acid. Anemia caused by blood loss occurs in hemorrhaged condition or in diseases like peptic ulceration, hookworm disease. Hemolytic anemia is caused by excessive destruction of the red blood cells. For example, in diseases like malaria or in hypervolemic condition, that is excessive water in the blood that causes lysis of the red blood cells. Aplastic anemia occurs from deficiency of hematopoiesis. It happens when the bone marrow is injured by cancers or from other disease tissues, metastasis, by autoimmune disease, or by intoxication from drugs like sulfurs and anticonvulsants, or by chemical substances like benzene, insecticide, paints, herbicides, solvents in general. Some genetic diseases also affect the bone marrow, causing aplastic anemia. What is the difference between white and red blood cells? What are leukocytes? Red blood cells are erythrocytes. White blood cells are leukocytes. Leukocytes are cells specialized in the defense of the body against agents, and they are part of the immune system. What are the types of leukocytes and how are they classified into granulocytes and agranulocytes? The types of leukocytes are lymphocytes, monocytes, neutrocytes, eosinophil, and basophil. Granulocytes are those whose cytoplasm are granules. When viewed under an electron microscope, neutrophils, eosinophils, and basophils are granulocytes. Agranulocytes are other leukocytes, lymphocytes, and monocytes. What is the genetic function of leukocytes? What are leukocytosis and leukopenia? Genetic function of leukocytes is to participate in the defense of the body against strange agents that penetrates it or made inside the body. Leukocytosis, leukopenia are clinical conditions in which the count of leukocytes in the blood sample is abnormal. When leukocyte count in the blood sample is above the normal level for the individual, leukocytosis is defined. When the leukocyte count is lower than the expected normal level, leukopenia is defined. The multiplication of this defense cell, leukocytosis, generally takes place when the body is suffering from infection or in cancers of these cells. Lowering these defense cells or leukopenia occurs when some disease attack cells like AIDS, when immunosuppressor drugs are used. In general, the body creates leukocytosis as a defense reaction when it is facing infectious or pathogenic agents. The clinical condition of leukocytosis is thus a sign of infection. Leukopenia occurs when there is deficiency in the production or excessive destruction of leukocytes, for example, in case of HIV infection. What are the mechanisms of hemorrhage contagion called? The physiological mechanism of hemorrhage contagion, one of them is blood clotting, are generically named hemostasis or homeostasis processes. What are platelets formed? What is the function of the platelets? What consequences does the clinical condition known as thrombocytopenia yield? Platelets are known as thrombocytes, are fragments of giant cells of the bone marrow called megakaryocytes. With their properties of aggregation and adhesiveness, they play a direct role in blood clotting, and they are also libellate substances that activate other hemostatic processes. Thrombocytopenia is a clinical condition in which the platelets count of the blood is lower than the normal. In this situation, the person becomes susceptible to hemorrhage. How does the organism understand that a clotting process must begin? When there are some tissues wounded with the injury of blood vessels, the platelets and the endothelial cells of the walls of the damaged vessels liberate substances, respectively platelets factors and tissue factors, that trigger clotting process. How can blood coagulation clotting process be described? Blood clotting encompasses sequence of chemical reactions whose respective products are enzymes that catalyze the following reactions. That is why the clotting reactions are called cascade reactions. In the plasma, thromboplastogen transforms into thromboplastin, a reaction triggered by the tissues and platelet factors liberated after injury of the blood vessels. Thromboplastin then catalyzes, along with calcium ions, the transformation of prothrombin into thrombin. Thrombin then catalyzes a reaction that produces fibrin from fibrinogen. Fibrin is an insoluble substance, precipitates to form a network and traps red blood cells and platelets, forming the blood clot containing hemorrhage. What are clotting factors? Clotting factors, these are substances, enzymes, reagents necessary for the clotting stage to happen. Besides these triggering factors, reagents already described, tissue and platelet factors, thromboplastinogen, thrombin, fibrinogen, calcium ions. Other substances participate in blood clotting process are clotting factors like factor 8, whose deficiency causes hemophilia A, factor 9, whose deficiency causes hemophilia B. What are the organs where most of the clotting factors are produced? What is the role of vitamin K in blood coagulation? Most of the clotting factors are produced in the liver. Vitamin K participates in the activation of several clotting factors, and it is fundamental for the well-functioning of the blood coagulation. What is factor 8? What is the genetic disease in which the factor is absent? Factor 8 has a function of activating factor 10 that in its turn is necessary for the transformation of prothrombin into thrombin in the clotting cascade. Hemophilia A is an X-linked genetic disease in which individuals do not produce factor 8 and so is more susceptible to severe hemorrhage. How is hemophilia treated? Why is hemophilia rare in females? Hemophilia is medically treated by administration of factor VIII in case of hemophilia A, of factor IX in case of hemophilia B, by means of blood and fresh frozen plasma transfusion. Hemophilia A and B are X-linked recessive inheritance, and for a girl to be hemophilic, it is necessary for both of her X chromosomes to be affected, while boys to have only one X chromosome are more easily affected. A girl with only one affected chromosome does not present the disease, since the normal gene on the unaffected other X chromosome produces the clotting factor. What is the epidemiological association between hemophilia and HIV infection? Since hemophilia patients need frequent transfusion of clotting factor 8 and 9, they are more susceptible to contamination by infectious agent present in the blood, from which the transfused elements come. In the past, blood bank did not usually perform HIV detection tests, and many hemophilic patients have become infected with the virus. What is anticoagulants? What are the practical applications of anticoagulants like heparin in medicine? Anticoagulants, these are substances that block the clotting reactions and thus stop the coagulation process. Ordinarily, there are anticoagulants circulating in the plasma, since under normal condition, blood must be kept fluid. In medicine, anticoagulants like heparin are used in surgeries in which tissue injuries made by the surgical act could trigger undesirable systematic blood clotting. They are also used to avoid the formation of thrombus inside the blood vessels of the patient facing increased thrombotic risk. What is discomor? How does the substance act in the clotting process? And what are some of the examples of its toxicity? Dicomor is an anticoagulant drug. Due to its molecular structure, dicomor competes with vitamin K for the binding site, blocking the formation of clotting factors and interrupting the making of prothrombin. Dicomor is found in some vegetables undergoing decomposition. It can cause severe internal hemorrhages when these vegetables are accidentally ingested. Coumarinic anticoagulant cannot be administered during pregnancy since they pass the placental barriers and can cause fetal hemorrhage. Streptokinesis is a substance used in the treatment of acute myocardial infarction. How does this substance act? A substance known as fibrinolytic, like streptokinesis and urokinase, can destroy thrombi, clots formed inside blood vessels, capillaries, or within the heart chamber, and are used in the treatment of obstructions of the coronary arteries and other blood vessels. Streptokinase destroys the fibrin network, and so it dissolves the thrombotic clot. Its name comes from after the bacteria that produces it, Streptococci. Thank you very much for participating in Blood. Please do like, share, and subscribe.